if you want to figure out how a really complex mechanical system works, say like a high-performance car engine or something,
Right.
a pretty straightforward method is to just take one part out and see what breaks.
Yeah.
Like you pull a spark plug, the engine misfires.
Yeah, exactly.
And so now okay, that's what a spark plug does. But you still don't truly understand the whole engine from that.
Yeah.
I mean, to understand how the parts actually relate to each other, the hidden dependencies, the structural redundancies, you have to start removing two parts at the very same time.
Right, because the single part approach, it really just gives you an inventory list.
Yeah.
But the two-part approach, that gives you a true wiring diagram. You start to see not just the components, but the fail-safes and like the parallel systems.
And that transition from a simple parts list to a dynamic wiring diagram is exactly what we're getting into today. So for you listening, we're doing a deep dive into this massive 2026 study published in the journal Cell.
A really incredible piece of work.
It really is. I mean, the researchers essentially took this exact mechanical logic and applied it to the human genome. They mapped the wiring diagram of a human cell by testing nearly 4 million gene pairs.
Yeah, the scale is just staggering.
It's wild. They systematically broke 2 genetic components at a time, just millions of times over, to discover roughly 89,000 hidden genetic interactions.
Which represents a monumental shift in functional genomics, honestly, because the mission of this research, it wasn't just, cataloging things.
Right.
It was to unpack this enormous genetic interaction network to figure out how genes functionally coordinate. And crucially, to explore how this exact blueprint can be used to uncover hidden gene functions, fight complex cancers, and even treat rare genetic diseases.
So, if you're listening, the main takeaway for this deep dive is about stepping away from the idea of the genome as just like a static sequence of letters in a textbook. We're looking at the complex, dynamic social network of our DNA. But I want to pause here because the sheer scale of this is, it's hard to wrap your head around. Testing 4 million gene pairs isn't something you just do with a couple of pipettes on a Tuesday afternoon.
Definitely not.
So let's look at the mechanics first. How did the cartographers actually build this map?
Well, the logistical foundation relies entirely on the CRISPR-Cas9 system.
Okay.
Specifically, they're utilizing something called the TKOV3 library. So they aren't just, splashing chemicals on cells in a petri dish. They are using guide RNAs as highly specific molecular scalpels.
Basically programming the scissors to cut exactly where they want.
Right, exactly. So the researchers introduced these guide RNAs into a vast population of cells. And each guide targets one specific gene and just shuts it off.
Right.
And by tracking the barcode of that specific guide RNA over time, they can measure the survival rate of the cells. Like, if a specific barcode just completely vanishes from the population after a few weeks, well, it means the cell with that specific knockout could not survive.
Okay, and doing that for one gene, I mean, that's fairly standard practice in biology now. But doing it for pairs means you are infecting these cells with 2 guide RNAs simultaneously, right?
Yes, exactly. You're creating a double mutant.
Which brings us to the core metric of the whole study, the genetic interaction itself. Because you're looking for an outcome that deviates from the simple math of just adding the single knockouts together.
Right. The deviation is the entire signal here. So a genetic interaction, or a GI, that happens when the fitness of the double mutant is significantly different from what you'd expect based on just combining the effects of the 2 individual mutations.
Okay, got it.
And we categorize these primarily into negative and positive interactions.
Right, and let's break those down. Negative interactions are often called synthetic lethality, right?
Yes, that's the term.
Which is such a cool name. But basically, this is when combining 2 mutations causes the cell to grow terribly or just outright die.
Yeah.
Even if the single mutations on their own were, mostly harmless. I tend to picture this like load-bearing architecture in a house. Say a biological process is supported by 2 parallel pathways. Let's call them pillar A and pillar B.
Good analogy.
You knock out pillar A, the cell's structural integrity shifts a bit, but pillar B takes the load. The cell survives.
Right.
You knock out pillar B alone, same result. The cell is fine. But you take out both simultaneously, and the entire metabolic roof basically caves in.
Yeah, the parallel pathway model is the classic mechanism for synthetic lethality. The cell has evolved these redundancies to protect really vital functions like DNA repair or cell division.
Because it can't afford for those to fail.
Right. So the negative interaction reveals those redundancies. But then you have the other side of the coin, right? The positive genetic interaction.
Specifically suppression, yeah.
Yes, suppression.
Yep.
Which reveals something entirely different. This is when the double mutant actually grows better than expected.
Wait, really? Better?
Yeah, it's fascinating. The second mutation somehow rescues the cell from the damage of the first mutation.
I've always found the concept of suppression totally wild. I mean, you break a second thing and the whole system improves. It feels counterintuitive.
It does, but mechanistically, it makes total sense if you think about cellular processes as like a flow of resources.
Okay, walk me through that.
Okay, say you have a mutation that breaks a downstream enzyme in a pathway.
Okay.
That causes a toxic buildup of some intermediate molecule. And the cell is essentially dying from the toxicity of that buildup.
Right, like a traffic jam.
Exactly.
Yeah.
Now, if your second mutation breaks the upstream enzyme, the one that actually produces that intermediate molecule, well, you've stopped the toxic buildup.
Oh, wow.
Yeah. You broke a second gene, sure, but you restore the overall biochemical balance.
Okay, that is a highly accurate way to visualize it. You're rebalancing the metabolic flux. So if the first mutation is like a leaky pipe that's flooding your basement, the second mutation is just going in and breaking the main water valve to the house.
That's a perfect way to put it, yeah.
Like, the plumbing system is definitely still compromised. You don't have water, but you've stopped the catastrophic flooding.
Exactly. You've halted the total system failure.
Now, I want to pivot for a second regarding the actual cells they used to run these 4 million tests.
Yeah.
Because they utilized the HAP1 cell line.
Yes, the HAP1 line.
Which is haploid, meaning it possesses only one set of chromosomes. But wait, let me push back here on behalf of the listener, because humans are, of course, diploid.
You're right. We have two sets.
We get one from mom, one from dad. So if we're using a haploid cell with only one set, aren't we studying something fundamentally broken or artificial? Why make that methodological choice?
It's a great question, and it comes down to efficiency and clarity. The signal-to-noise ratio in a haploid screen is vastly superior to a diploid one.
How so?
Well, in a diploid cell, if you knock out one allele, one copy of the gene, the second allele on the other chromosome often upregulates to compensate for the loss.
Ah, so it masks the effect.
Exactly. It completely muddies your data. By actively choosing the HAP1 haploid line, they completely stripped away that diploid backup copy. The researchers ensured a pure unmasked readout. When they knocked out a gene, it was truly gone.
No safety net.
None. And this efficiency is exactly what allowed them to scale up to 4 million pairwise tests. Without that, the data would have just collapsed under its own noise.
Okay, that makes total sense. So just to recap the mechanics for you listening, they run this massive CRISPR screen. They sequence the barcodes, they calculate the fitness scores of the cells, and they identify about 89,000 of these positive and negative interactions. They have their raw wiring diagram.
Right, the raw data.
But raw data isn't knowledge. I mean, just having a list of connections doesn't tell you how a city works. The real breakthrough in this paper is how that massive web of connections actually organizes itself.
It's not a random hairball.
Right. It forms this highly structured hierarchy.
And the organizational principle they discovered there relies on something called genetic interaction profile similarity.
Okay, unpack that term for us.
Yeah.
Profile similarity.
So instead of just looking at whether gene A simply interacts with gene B, the researchers looked at the entire interaction profile of a single gene. They mapped out every single positive and negative interaction a gene had across all those thousands of tests.
So they basically built a social media profile for every single gene.
Essentially, yes. That's a great way to think about it. And then they used mathematical correlation, specifically purest correlation coefficients, to compare these profiles against each other.
Like mashing algorithms.
Right. If two genes share a highly similar pattern of interactions, meaning, they are lethal with the exact same partners and they're suppressed by the exact same partners, it strongly implies they function together.
They must be in the same physical complex or the same biological pathway.
Exactly.
It's basically the ultimate algorithmic guilt by association. Like, if gene X and gene Y both cause the cell to die when they're paired with the exact same set of DNA repair genes, you can confidently bet your money that gene X and gene Y are also involved in DNA repair.
Yes. And when they applied this clustering logic across the entire map, it revealed this really beautiful nested architecture, sort of like Russian nesting dolls.
Right. I love this part. Walk us through the levels of those dolls.
So the topology is incredibly clean. At the most granular level, the bottom level, the profile similarities grouped genes into 71 specific protein complexes.
And these are proteins that literally bind together to form cellular machines, right?
Exactly. Then those 71 complexes nested perfectly into 17 broader biological processes, things like vesicle trafficking or RNA processing.
Okay.
And then scaling up again, those 17 processes map neatly into 7 major cellular compartments,
Wait.
like the mitochondria or the nucleus or the endoplasmic reticulum.
It really is a functional map of the cellular city, but, just like any city, not all the roads or buildings are equally important. And the map highlighted this critical subset of genes, the hub genes. Hubs. Yeah, the top 5% most connected genes in this network. They exhibited roughly 6 times more interactions than your average run-of-the-mill gene.
Hub genes are the foundational infrastructure of the cell. And the researchers noted they share some really distinct physiological traits.
Like what?
For one, they're highly expressed, meaning the cell invests a massive amount of energy just keeping them active all the time.
Makes sense if they're hubs.
Right. And they also tend to localize in constitutive anchor regions of the genome.
Okay, constitutive anchor regions. What does that mean in plain English?
Basically, these are physically open, highly accessible zones of chromatin that are just constantly being transcribed. The DNA is unwound and ready for action.
Always open for business. And they also found these hubs are evolutionarily much older than the less connected genes, right?
They did, yes.
Which makes perfect mechanistically sense to me. The oldest genes built the initial foundation of the eukaryotic cell 1 billion years ago. Every new evolutionary adaptation over time had to integrate with that existing foundation.
Right, which naturally turns those ancient genes into central hubs as things build around them.
Right, and this structural understanding, this guilt by association, it actually offers some immediate practical utility for the listener.
Oh, absolutely. Because of this nested hierarchy, the researchers were able to confidently assign biological functions to 113 poorly characterized human genes.
Wow. Mystery genes?
Exactly. These were genes we had sequenced years ago. We knew the letters, but we had absolutely no idea what they actually did in the body. But by simply observing which specific neighborhood they clustered into on this new interaction map, their roles became obvious.
That alone is a massive leap forward for biology. I mean, we've taken 113 mystery parts from the inventory list and finally put them in the correct section of the engine manual.
It's huge.
But, exploring this human wiring diagrams is really only half the story in this paper. To truly understand the architecture, the researchers took this brand new human map and compared it to a genetic map made years ago for a fundamentally different organism.
Budding yeast, Saccharomyces cerevisiae.
Right. And the comparative genomics aspect of this study is arguably its most profound basic science finding. I mean, we are talking about 2 species that diverged from a common ancestor over 1 billion years ago.
1 billion years of separate evolution.
Which is exactly why my initial reaction to this part of the paper was, frankly, high skepticism. I mean, 1 billion years. Adapting to completely different environments, scaling from a microscopic single cell to a highly complex multicellular mammal. You would naturally assume the genetic wiring would be completely unrecognizable by now.
You would think so. And honestly, if you look at the highly specific individual gene-to-gene interactions, just the one-to-one specific pairs, you do see that massive divergence.
Okay, so the specific wires changed.
Right. Only a modest fraction of those exact pairs are conserved between yeast and humans. But the researchers didn't just look at individual wires. They zoomed out and looked at the global topology.
The structural rules.
Exactly. The overarching structural rules of the network are remarkably conserved.
I compare this to the underlying logic of a 1920s telegraph network versus a modern internet routing system.
That's interesting.
Right, like the physical wires are totally different, the exact endpoints are different, and the modern internet has millions of new, highly localized connections. But the foundational rules of how a signal is routed, how hubs distribute information, and where you place redundancies to prevent a total system failure, that underlying topology hasn't changed.
That's a perfect analogy. And the data in the paper bears that out beautifully. When they measured the density of negative interactions within specific core processes, such as, say, DNA replication or how the cell manages essential metabolites, the structural density was nearly identical.
Between a fungus and a human cell.
Exactly. The network density was the same.
Which tells us that nature is incredibly conservative when it comes to the core operating system.
Very conservative.
Once evolution locked in an architecture that could efficiently and stably manage the immense complexity of eukaryotic life, it just didn't mess with it. It built on top of it, sure, but it didn't change the foundation.
Right. And the implication for modern medical research here is just vital. We constantly rely on simpler model organisms like yeast or fruit flies or nematodes to study human disease.
And sometimes people wonder, why are we studying flies to cure human cancer?
Exactly. But this billion-year conservation of network topology, it rigorously validates that approach. It proves that we can manipulate a pathway in a simple yeast cell, figure out the network-level behavior, and then confidently predict how that same pathway will behave in complex human biology.
It totally bridges the gap between basic evolutionary biology and applied medical research.
It does.
And that transition actually brings us to the real crescendo of this deep dive.
Yeah.
Because understanding a billion-year-old structural blueprint is intellectually fascinating. But the researchers didn't stop there. They immediately turned this map into a weapon against disease.
Yes. To do that, they had to integrate their new data with an existing incredibly important resource called the Cancer Dependency Map, or DepMap for short.
DepMap.
DepMap is this massive database cataloging essential genes across hundreds of different cancer cell lines, each with its own unique genetic mutations.
Okay, so let's clarify the difference for the listener. DepMap is looking at single genes across a wide variety of genetic backgrounds to see what a specific tumor relies on to survive.
Right.
Whereas our new HAP1 map is looking at pairs of genes in a single controlled background to find hidden interactions.
Exactly. There are two different scales of data, and integrating them requires some heavy computational lifting. So the researchers utilized a deep learning algorithm called Bionic.
Bionic.
Yeah. And by fusing the pairwise interaction data from the HAP1 map with the broad contextual data from DepMap, the algorithm could predict genetic dependencies in cancers that neither dataset could actually see on its own.
That's amazing. Let's ground this in a specific clinical mechanism so everyone can visualize it. Let's look at cancer first, specifically utilizing those negative genetic interactions.
Synthetic lethality.
Right, the synthetic lethality we discussed earlier, where taking out two pillars causes a collapse.
Okay, so the paper highlights a profound negative interaction between a gene called PILO and the SKI mRNA surveillance complex.
PILO and SKI.
Right. The mapping data showed that if you knock out both of these at the same time, the cell suffers a catastrophic translational failure and dies.
Okay, but how does that help a patient in a hospital? Because a healthy cell in your body has both of those functional. If you give a patient a drug that targets the PILO gene, their healthy cells will just lean on the SKI complex. They survive.
True. But the therapeutic window opens up because cancer cells are notoriously genetically unstable. They have missing pieces. Many specific cancers, like certain glioblastomas or melanomas, carry large structural deletions in their genome.
Okay.
And one very common deletion is known as the 9p21.3 deletion.
9p21.3.
Yes. This chunk of missing DNA often takes out tumor suppressor genes, which is bad, right? That helps the cancer grow. But it also takes out passenger genes that just happen to live in that exact same neighborhood.
Ah, guilt by location. So the deletion is a double-edged sword for the tumor.
Exactly the point. The 9p21.3 deletion often takes out critical components of that SKI complex.
Oh.
Right. So the cancer cell is already structurally compromised before we even do anything. It has lost pillar A. It is surviving entirely because it still has pillar B, the PILO gene, to manage ribosome rescue and mRNA surveillance.
Okay, I see where this is going. So the tumor is uniquely vulnerable in a way the rest of the patient's body is not.
Yes.
The healthy cells have both pathways intact. The tumor only has PILO. So if you, as a researcher, develop a targeted inhibitor drug for the PILO gene, you administer it. The healthy cells absorb the hit, they switch over to SKI, and keep functioning fine.
Right.
But the tumor undergoes a catastrophic synthetic lethal collapse because you knocked out its only remaining pillar. You've targeted the specific hidden dependency.
It is the holy grail of targeted oncology. You aren't just indiscriminately poisoning rapidly dividing cells with chemotherapy. You are exploiting the tumor's specific genetic architecture against itself.
It is brilliantly elegant. I love that.
It's amazing.
Now, let's look at the other side of the therapeutic coin. We've used negative interactions to kill cancer cells. How are they using positive interaction suppression to save cells?
Well, suppression is uniquely suited for treating rare inherited genetic disorders.
Okay.
In the paper, the researchers explored Barth syndrome. This is a really severe, often fatal disease characterized by cardiac and skeletal muscle weakness. And it's caused by a loss-of-function mutation in a single gene called tafazin.
Tafazin. And mechanistically, tafazin is crucial for the mitochondria, right? The power plants of the cell.
Exactly.
It helps modify a specific lipid called cardiolipin, which is basically essential for the mitochondrial membrane to fold properly and produce energy. So a tafazin mutation means the cellular power plants are fundamentally broken.
The metabolic engine just stalls out, yes. Now, traditionally, you might think the only way to cure this is through direct gene therapy, like physically trying to insert a brand new healthy copy of the tafazin gene into the patient.
Which is incredibly difficult to do safely and effectively.
Highly difficult. But the genetic interaction map revealed a hidden workaround. It showed a strong positive suppression interaction between tafazin and a totally different, seemingly unrelated gene called ABHD18.
Wait, so the map suggests that if a patient has a broken tafazin gene, actively breaking the ABHD18 as well will somehow make things better.
The interaction profile strongly predicted it, and then the researchers went into the lab and validated it biologically. When they knocked out ABHD18 in cells that were actively failing due to the tafazin mutation, the mitochondrial fitness was remarkably rescued.
Mechanistically, how is a second knockout rescuing the mitochondria? Is it slowing down a competing pathway to balance the flux, like the leaky pipe analogy?
Yes, exactly. While the exact molecular gymnastics are still being fully unraveled, the principle of suppression here likely involves altering the mitochondrial membrane dynamics or the lipid metabolism pathways to compensate for the lack of mature cardiolipin.
Interesting.
By removing ABHD18, the cell is basically forced to reroute its metabolic plumbing, and that new route just happens to bypass the bottleneck created by the original tafazin defect.
It's an incredible paradigm shift. I mean, we've spent decades looking at genetic diseases by staring only at the broken gene, right? Just agonizing over how to fix that one specific broken part.
Always zooming in.
But this wiring diagram proves we should be zooming out, looking at the entire neighborhood. We shouldn't just ask, how do we fix this one gear? We should ask, what other lever in the machine can we pull to bypass this gear entirely?
It completely transforms incurable monogenic diseases into network routing problems. If we can find the right suppressor gene on the map, we can design small molecule drugs to inhibit it.
Essentially triggering the biological workaround pharmacologically, just taking a pill to reroute the network.
Exactly.
It really reframes the entire concept of a genetic vulnerability. So for you listening, we've covered massive ground here today. Let's do a quick recap. We started with the sheer logistical scale of mapping 4 million gene pairs using CRISPR and those unmasked haploid cells.
Yep.
We saw how those 89,000 discovered interactions organized themselves through profile similarity into a beautiful, nested hierarchy of complexes and biological processes.
The Russian dolls.
Right. We looked at the profound billion-year conservation of this topology from yeast all the way to humans. And finally, we explored how fusing this map with DepMap and deep learning turns these abstract connections into literal treasure maps, finding the synthetic lethal Achilles heel for cancer and the functional suppressor workarounds for rare diseases like Barth syndrome.
The integration of all these methodologies, the CRISPR screening, the comparative genomics, the machine learning, it makes this study a true cornerstone for the next decade of functional biology.
It really does. But the detail that truly stopped me in my tracks, and the thought I want to leave you, the listener, with today, is the sheer scale of what is still undiscovered.
Oh, absolutely.
Because this study was a colossal undertaking. Nearly 4 million gene pairs tested. Yet, statistically, they have currently mapped less than 5% of the total possible human gene pairs.
Barely scratching the surface.
The researchers estimate the complete comprehensive wiring diagram of the human cell contains roughly 1.4 million interactions.
Yeah.
We are currently looking at a map with 95% of the territory still shrouded in total darkness. Think about what that means for the future of medicine. As we map that remaining 95%, we are moving entirely away from simply reading our DNA sequence and moving toward truly understanding the butterfly effect of every single genetic variation in our bodies.
It's a whole new frontier.
It is. So, my question to you listening is this. When we finally have that complete 1.4 million interaction map illuminated, when we can see the mechanism of every single hidden wire, every redundancy, and every suppressor in the cellular engine, will we reach a point where where there is no such thing as an inherently incurable genetic disease.
Wow.
Will every disease simply become a network pathway we just haven't successfully rerouted yet? Just a stalled engine waiting for a mechanic who finally has the complete manual.
That is the ultimate goal.
Something to think about as we keep exploring. Thanks for joining us on this deep dive.